So, hello, good morning. It's slightly early, but I wanted to know a little bit about you. Start. So, because I haven't read the applications of the details, and I don't remember. How many of you are all of you from? No, are from biology. From biology. And how many undergraduate students? Graduate. So maybe every now and then I will go a little to basic and more sophisticated. And so I will try to go around with different types of. I will talk about biology. Please, biologists, forgive me if I say something that's good. Anyway, I'm a physicist. I work on. So, starting with the itself, I wanted to describe briefly how we are planning to organize it. So the other professors here are Rafael Meneses and Pablo de Castro. We are going to give 3 lectures. The agenda here is the website of the and we had also hands-on activities. They are planned. So they are planned with Wednesday. There will be, and we have a reserved space with computers, but I think that we might stay here and so you work with your own computer. And they did that we divide you up, count it, but 38. But we are going to divide you, and all of that is going to have that organization. And so we're talking about tracks, like that briefly on the web, but very recently. I don't know if you read that or not. So one track is Pablo's course, track is Rafael's, another is mine, and each of us will. And once you pick the track, you that track. We're good. And so you follow the hands-on activity of that track, group separately. There is that some assignment, then that depends on each of us. In my case, it's going to some parts of books that I would like you to understand and maybe patience, and then expect you talk, 25-minute talks per group on Friday. And thought here for you to hear the presentation. Of course, how is done. Okay, so let me start. Oh, here is the group. Friday is the. Okay, so I am going to cover some. I share where yeah collabs. If this goes to the parent folder or directly to apps, there is also the guides with my track. And what we are going to focus on in this part is processes that occur inside cells. I will show some experimental results, but the focus on model. What we are going to look at sites, looking at what happens inside cells. Look at some of the mathematics. They are old-fashioned, basic principles. And now have much time. Yeah. Of course, everybody that is interested. There are some nice books. They are course loosely based, much longer. Were the books that I got inspiration won't use. But some of the exercises I stole them from one. And other physicists, logical, they keep very interesting site that is called biology by the number, cell by the number. Exercises are going those now. Part of the book starts with getting a size, the molecule, what's the size of an organ. That we are going to use. The physical models, very interesting. And from there, I am going to take all the examples for the groups that pick this track to make the presentation. And a little something about the smallest version, Albert's book, Molecular Biology, is this biology. Okay, so basically all living systems are that could be prokaryotic, eukaryotic cells, or most. Eukaryotic cells. And look, and let me give you a little bit of jargon. I will do that during the course. A lot of detail, but just language. So the cytoplasm that is not eukaryotic cytoplasm there. The cytoplasm that is outside is cytosol. So then the plasma membrane, the cell membrane, but the cells cannot be get nutrients, but they also need. So in order to exchange this, they are sensing environment, and they generate responses. They react, and on many occasions, are manifested as changes in concentrations. Some substance called ligands, ligands, because cells detect those changes through binding. Ligands bind some stuff that is on the cell membrane, and that induces. So this is the and here there is a plasma membrane, the ligand side binds here, and it have some parts that are outside and parts that are inside. And through binding something outside, they can change something inside, and that can propagate other changes. the change use changes called a signaling process that occurs. And that's what the aim of this course, we are going to cover very basic things. Then the last day, we are look a little bit at the signaling cascade. Okay, so to sense what happens, the signal needs to cross the membrane. So there are different ways of doing crossing. One is that one that I showed in that picture, right? Receptor that cross parts, and some of them coupled. They are coupled some proteins by this. So uh, when the ligand binds, there is a conformation change. That part of what found receptor. So they can activate, then activation further changes in a propagator. Another way of transporting, crossing this membrane, for example, an example that I'm going here is how neurons communicate. But there are different ways. One way, which is chemical synapse, and it's communicate with muscles. And neurons are what are called excitable. So another so you have a little bit voltage difference. So this changed from the soma of the. So that cross are sensitive to the voltage. Change the calcium. Changes in the. So basically, the other case change binding. So what we see basically binding, transport, also calcium get to the plasma that they we are going binding and unbinding scale. Go a little bit types of molecules, and also because some of the the composition of cells. Water. Energy. Permanent. But some units that we are try because but the thermal for a 25-degree centigrade much more than those numbers, so they are very strong anyway. So cells are compounds that contain carbon, oxygen, and 99% of their cell mass. And if we water, well, all molecules carbon, and so the processes that occur in organisms are a consequence of the competition, stability of the bonds carbon atoms form, and the ability of some catalysts are called rearrange. And also the properties of water. And in particular, the polar nature of water, by this that it has dipolar moment. So it's a dipole. And the oxygen have a dipole. So that has gives a particular characteristic. Has so much. And also the way that water induces a lot of the, or affects a lot the behavior itself. Well, water makes a lot of them. One of the things that happen with water is that the molecules tend to form net. And so they are connected, hydrogen. They are rearranging all the time. So and because of the polar nature, you have all these, when there are ions in there, the water molecules organize them. And also other polar molecules. And so there are substances that are polar, also are either charged or have a dipole that can fit within this network that the molecules of water form. And those are called and those molecules that don't have that, they disrupt hydrogen. So they are hydrophobic. Get well inside that net. Now, this part is related to these calculations molecule, part of the first sense of how many molecules we have in the typical length scales of a cell. Particular, the length of a molecule is of the order of nanometer. So in any macroscopic volume of centimeters, have many, many, many molecules. So one of the ways in which you count molecules, produce types of units are not so common, but they are very common Instead of counting them one by one, count them in terms of how many Avogadro number of molecules we have. And that's what is called one more molecules number. So, but when you think of cells, mole, very large. Problems. But cells that typically are 100 microns. We have a 10 micron, let's think a cube, 10 micron at the size. Then when you compare moles, have given that the size of the molecule would that's of the order. So usually you not moles, but some that are small. And something that is important usually when we describe how much we have of a given substance in a cell, we think of concentration rather than number of molecules or of moles. And which is the number of molecules. Now, usually units that not 1 over a units of length cubed, common thing. In terms of what is called, which is measuring volume. So, and very large. Cell, well, this concentration. But other cell components. So if we protein here are given in terms of their mass, consider what's the typical mass protein in types of units mass that what you see that you have example of large, but yet not quite. So now let me a little bit of names, some of the molecules that are inside cells, and have many macromolecules, proteins, also nucleic acid, that is and we also have what I call molecules, they are polymers, where they have different types of molecules. Mainly the first are the energy. They also like joining molecules, or many of them are basically other types of molecules acid that form, hopefully, the that form the hydrophilic end likes to contact water, hydrophobic. So when putting water, they are organized layers by layer, which the hydrophobic tails get one in front of the other, heads which hydrophilic are in contact side of the cell or then the amino particular type of structure. There are 20 proteins. So proteins are macromolecules that are formed. One talks about different parts of the structure of a protein, primary structure, which is sequence of we are going to, but how you produce the proteins information in DNA and sequence, and you translate that in that sequence. Primary. Then once they fold in a three-dimensional object, there are some that are very proteins, secondary structures. Some of them are called alpha helix, others are and finally quaternary structure. Many times the proteins like substructure, subunits. So this would be subunit. Many of them arranged in something more. Then the nucleotides, taking form the acids. How they are particular forming this double helix. And you have the double helix, you have single strand RNA. Go from the genetic eventually the energy using phosphate, triphosphate, diphosphate, large amount. The reverse process transfer energy from side favor description. That was a little bit try to describe in actions. That was a presentation of components, molecules, structures without going in much detail, and now we are going. So the chemical usually or many goals that to establish two molecules for the encounter. You need the molecules first in. Usually that's done in when you're in aqueous. Happens. But this transport that allows the molecules to find one Pablo is try to. Now, once they encounter, once they are bonds range, jumping landscape. And minimum go through the barrier, then that's done. Now the how easy cross the relationship barrier. So the idea of a mechanical system, there is you're in a potential, but you kicks particle, and you give kicks, and suddenly one of the allows to this. Eh, eh, that way. Big molecule. That's that's type of reaction. And the thermal probability jumping from height so it can if the barrier is long time. And have something that has like two parts that can come apart. conformation, like the one that we saw with higher now sometimes talk about this don't all the processing traffic sticking with you're not including that in the path. So we talk about the probability. Okay. So we are going what we are operations in chemical. And so let's suppose that we that can join together. Then we are going. Now for A and B, N our volume, small volume within which the two molecules have to, so that they can decide whether they are close enough together to be able to. Anyway, so what I want to now is suppose that I have and I divide them volume so that on in each of these small volumes I will molecule that is equal to Na or volumes. And this is okay. This is how many molecules equivalent of are in the stream. And so I am here. So I can form, let us assume that this is certain. Then we will see the description. So I can so that's couples I can form between. When they come together, they decide. And now they come together and they decide during a certain time because all of them are moving, so there is like a certain time they remain for moving out. So let me and so we of the probability of given that they met, the probability that they decide to join. So events we join something times ones that I forming C times probability per unit. No, the longer it's the way in which okay. And then if I have this possibility joining A and B to form C, I also that comes apart, forms back B. Process with another probability. Joining C in volume, little volume. And inside the little volume, so during a time delta t, the number of C that will come apart and go back to A plus B separate, those will the time I'm here this. First, they are how often they are together in a little volume where they can interact. Then I look at the time, which is the typical time they stay, and then I see many reactions I one direction or the other inside. And I don't want to count all the reactions. So these are the number of reactions that occur, delta T, inside on average, inside the little b. And so if I look at the whole volume, number of reactions delta. But what I have to do is multiply by how many of the small v volumes I have. That number n times the and the number of reactions large V. The number of these volumes. And so what I can think of is, I know how many reactions. I can compute how many molecules A, how it changes over the whole. So each time I have a reaction, A plus goes to, I decrease the number of molecules A, volume, by 1. And I increase. So I can compute a number if I look at a certain time and a time immediately after, I differs from the previous by this time at which I. So what I just said that A will by 1 per reaction, 1 molecule per reaction, I have this. By 1 molecule per reaction, each time I have the inverse one. So here I have the reactions that go from left to right. And the ones I have from right to left. Yes. This is the probability that once you have them together, they can bind. Now we are going to the probability of the molecules together in a particular way, because here I am like counting molecules, as if in those little volumes I had many. But usually you have less than 1 in the interaction. So instead of counting molecules, that may be the argument, you will use probability, then it's that product. Having both. But I'm going to that after. Okay. So then what I have going to and I'm going to divide also delta. And I have when I do NA, but if I look at A, little e, was big, and an concentration. Put a little b here so that I get a concentration. So what I have over the times the limit delta t. The difference between the concentration at the same time, that is infinitesimal distance divided by time and reaction. What you see that time yes, each goes to 1A and okay, and so concentration A was so great. And as I told you, we did this a little bit hands. thinking that we have many molecules by the small volume, but usually the situation have a dilute. All of this volume is filled with water, and the molecules are there, diffusing, bumping against the water molecules, and they usually are in very small, so that small concentration inside the volume, which is of the typical size where they can react. If I look at those distances, I don't have a stellium, very few. And so these numbers actually are usually one. Every now and then I find in one of those little V's one molecule, but very rarely I will find another. And so these numbers, these small numbers, I think that they are between 0 and 1. On average, they are actually. So that this is a calculation. I can also do, instead of thinking of counting pairs, I can think that those ratios that I put here actually correspond to the probability of a molecule. So this is the probability of one a molecule. So this, what I thought of as how many pairs I could form, the molecules, this would now be the product probability of finding a molecule and E molecule. That's the probability. The joint probability of finding A and B molecule. In the so this way of describing reaction, action, action, and you have other reactions, molecules. And in this case, now you would have to, for instead of couples, product of probability, I have to encounter 3 molecules. So this is related to the probability of being together. One B. Now I have to two ways, and so if they are all independent, they find the ways B E will be A, A, B, B. A's. So when I count ways of reaction, here I will concentrate square. But on the other hand, when I count the reactions, I want to count how many molecules 8 Freud in this A type. Each time I have a reaction, way, how how many molecules. And when I go into, so this two here has two for the reaction. One is, and the other one is that I ate. So that if I have while of reaction same, but each time I do the same way. See, let us assume a reaction alpha. Some of these, so for general form of writing, then write the equation. One hand counter of all of these. Notice that if and then on the other side, I would also say K, the number of molecules, one, here of the encounter. Then K of the transition probability joining action. Going from the right to the left, and then I have to count how many molecules I destroy or create through this. When I go from a left to side, right, destroy alpha 1 molecule, B1, 8 beta 1, so that this will be one. Okay, so this is mass action. What it says is that equilibrium negative is 0. All of this product, this product is equal to this product there, equivalently. one. That's it. Yeah, because it's related to the relation, the probability height of the barrier, and okay. So a summary here, and let me tell you something. Sometimes the actions approach of together, then we, and so when things are diffused, I don't know, Pablo with that, yeah, certain volume here, by that quantity, then the quantity determines a little bit the k-on dependent. Okay. So, okay. So we said that in order to overcome a barrier, just temperature take a very long time, and because you have to rely part of the molecule going fast and easy, very, that's the probability that those probabilities may go from barrier and very long. So it can take very long, and what times. And here is how enzyme. Here what we are thinking of A plus B that will done the help. There is something represent substrate. And what you see is that substrate binds something that when there that little hole, the enzyme helps the substrate become product. We arrange so then that's this. And the way in which that is done is that this additional binding that you lower the arrangement. So go back at this. The first model of was the ways simplest. Simplest. What we have goes to complex. Well, and once you are in the rate, the reverse for explanations. All the elementary reactions, one of them probability, right? And you know how rare that very small. The other possibility happens that once you have the product, you move it somewhere else to, so there is no. But this is the Michaelis method. And we can now that we know mass action of the invariant upstream and the substrate will disappear. Reaction back. Representing reaction. These are like two reactions written in the same line. Then if I go I have the same reaction. But also is here comes from producing. One hand it goes like once you create to destroy an vice versa. Here when you so one thing that we see then you that if you add up they can and that some of the time derivative, what it means constant. So people, I mean, we could have deduced that from the starting. This means enzyme can be free without having any bound to it, or it can be with the substrate bound to it. But it never disappears. And so the total amount has to be fixed, either as complex or as a free enzyme molecule. That's it. So this means that I can get simply replacing E by the constant that I minus this remains. This and the equation. So let me equation for S, E, and C, and I forgot to write the equation for the product because we are after at what rate product is involved in only one reaction. The rate, the product is the velocity, and so what we are after. And one way past it was binding was much faster than the rate. So that case, so you have this jet ski goes very fast to first month. So it's like I forget about this. I could forget about this. Equation, this reaction, would go to equilibrium. So what Michaelis said is, because of this time scale separation, I can assume that this reaction here is like in equilibrium. Basically forget. So they are in a and so this term is equal to this term. And 1. Which I can write 1 and what I have here, 8 over units of. Why? So K2 is small. I produce the product at a small rate, which means I deplete the substrate at a small rate. So it more or less remains constant for a long time, so that I can think that this S is dot velocity. And what I see for S goes to A2. And why this is K over 2 when where maximum. Well, this is the Michaelis approach, but there is more sophisticated way of but basic called the quasi. The idea here, is the concentration of much smaller substrate. I introduce very small. And if I do that, then fine. If I do that, with those what I see I want to compare equations because they have a bunch of things that are very similar. But here I have what I see is that this term here, but in this equation, y, which is and these terms are similar, but this one has an s going to put I by. So it's similar. These two terms are similar, but this one has this. So they have terms that are actually exactly the same, exception of the last one. But the main difference is that substrate has those terms multiplied by a very small. And this one here doesn't want to go through the presentation. I hope that you understand. Do it a little fast. So what you have variation epsilon times some term, while the variation has those terms without the epsilon. So this variable evolves on a faster time scale than this one. This one evolves slowly. So S over S0, which is and 0, X fast. And so for this one, like 6 constant because it evolves so slowly that it's IC constant. So X on this fast time scale follows the slow changes of sigma. So it's like this goes to equilibrium, X goes to its own equilibrium that depends on very fast. X like goes to an equilibrium that is changing slowly. That's what is called the quasi-steady state. One of the variables varies very slow. The other one follows those variations, adjusting to the new value on the fast time. The idea is this, and the result is similar. The Ellis-Menten one is the scales. But here, instead of okay, so when because see, this is a way plotting straight line, and there are other ways we're going to ask, which is called Actually, enzymes are large molecules, have many bindings, and you can bind the substrate more than one of those. And sometimes what happens is that bind the sites. Now binding comes was the first binding produced. And if that happens, that the binding favors binding, then you talk about the beginning, not easy to bind, but then once you bind one substrate molecule, the second one, the dependence of the velocity that reflects that we are going to instead of so that if the substrate concentration is low, but once you pick up enough binding time, then the next one is very easy. The dependence molecule is some scales like instead of that comes here comes anymore you bound. Okay, we are going to finish now. Other enzyme mode. We'll move to okay. Today in the afternoon, Pablo. Sí.